Hey, what's up everybody, it's Roy from India. Welcome to my channel Roy's Academy. Make sure you watch the video to the end, because if you don't, there might be some punchlines at the end of the video which is very much important for clearing the concept. And also at the end of watching the entire video, please do hit the thumbs up button and also comment down in the comment section below and let me know if you have any personal queries or doubts. And also you can show me your love and affection by subscribing my channel, so please do subscribe my channel and help me grow my channel more and more for you people, so that I can help benefit you people with more such videos. Thank you. Okay, so now in case of the pyrimidine metabolism, the important amino acid that takes part is the glutamine. Okay, the glutamine is nothing but acidic amino acid, the glutamic acid, as we all know. This glutamine, along with two other molecules, one is the CO2 and another one is ATP, adenosine triphosphate, which provides energy for the conversion. Okay, now this ATP molecule here needed is 2 ATP molecules along with 1 molecule of CO2 and 1 molecule of glutamine. Now all this compound, in the presence of the enzyme carbamyl phosphate synthetase 2, in the presence of enzyme carbamyl phosphate synthetase 2, gets converted to what? Carbamyl phosphate. Carbamyl phosphate. So here the glutamine is playing a major or vital role. Why? Because the 1 molecule of glutamine plus 1 molecule of CO2 plus 2 molecules of ATP, which provides energy for the conversion of glutamine to the carbamyl phosphate in presence of carbamyl phosphate synthetase 2 or type 2 enzyme. But why the glutamine? Because glutamine is that basic amino acid, which helps in the formation of the pyrimidine nucleotides, or helps with the pyrimidine metabolism. Okay, so unless and until glutamic acid or glutamine is being synthesized or produced as a byproduct in our body, the conversion of glutamine to the pyrimidine nucleotides, to the de novo pathways, is not possible somewhat. Now this carbamyl phosphate group, in the presence of aspartate carbamyl transferase, carbamyl phosphate in the presence of aspartate carbamyl transferase, gets converted to what? N-aspartate, N-carbamyl aspartate, N-carbamyl aspartate. This is what it gets converted to. Okay, in the presence of aspartate carbamyl transferase, along with liberation of one PI molecule, one inorganic phosphate molecule, this entire conversion takes place. Now this N-carbamyl aspartate, in the presence of another enzyme called dihydroorotase, gets converted to dihydroorotate. Okay. Now this dihydroorotate, again in the presence of dihydroorotate dehydrogenase, gets converted to orotate. Orotate. Here the conversion takes place with the donation of one proton from the dihydroorotate. Dihydro means two hydrogen atoms are present, right? So from these two hydrogen atoms, one hydrogen atom is being liberated or donated. That donated hydrogen atom is being taken by NADH plus H plus from NAD plus. Now this orotate, in the presence of orotate phosphoriboside, orotate phosphoriboside transferase, phosphoriboside transferase gets converted to the product orotidine. Along with the liberation of pyrophosphate. The orotate helps in the liberation of the pyrophosphate. From orotate, the pyrophosphate gets liberated, this compound. And in the presence of orotate phosphoriboside transferase, it's converted to orotidine. So orotidine, this compound, is the first stable pyrimidine base compound or basic compound. From this orotidine compound, the other pyrimidine bases that are necessary, the pyrimidine like the uracil, uridine, thymidine or the cytidine bases are being formed. So orotidine is the first basic stable pyrimidine nucleotide base or pyrimidine class of nucleotide. Now from orotidine, this phosphorylase, orotidine phosphorylase helps in the formation of, in the presence of enzyme, orotidine forms orotidine 5 monophosphate. This is OMP. OMP is nothing but orotidine 5 monophosphate. Now this orotidine 5 monophosphate, in the presence of OMP decarboxylase gets converted to uridine 5 monophosphate. Uridine 5 monophosphate, UMP. This is orotidine 5 monophosphate, and this is uridine 5 monophosphate. Now this uridine 5 monophosphate, in the presence of UMP kinase, uridine 5 monophosphate kinase, along with the help of ATP to ADP conversion, ATP to ADP plus PI. So ATP is broken down to ADP plus PI. This energy is taken by UMP kinase to convert what? To convert this uridine 5 monophosphate to uridine diphosphate. Now this uridine diphosphate, in the presence of again the same procedure, UDP kinase, and with the conversion of ATP to ADP plus PI forms uridine 5 uridine triphosphate. Uridine 5 triphosphate. Okay, now this uridine 5 triphosphate, in the presence of enzyme CTP synthetase, cytidine triphosphate synthetase, gets converted to what? Cytidine 5 triphosphate. Cytidine 5 triphosphate, and that too in the presence of ATP, which gets converted to ADP plus PI. So this energy ATP gets converted to ADP plus PI is being utilized by whom? CTP synthetase. So CTP synthetase utilizes ATP to ADP plus PI and gets converted to cytidine 5 triphosphate. So you can see that this cytidine CTP, this compound cytidine triphosphate, uridine triphosphate, these two are the stable pyrimidine compounds or the pyrimidine class of nucleotides that are being synthesized in the pyrimidine metabolism part. Unlike the purine metabolism part, where the adenine 5 monophosphate and the guanine 5 monophosphate, AMP, GMPs were all of them being synthesized. So uridine triphosphate or uridine diphosphate, monophosphate, cytidine triphosphate, these are uridine, cytidine, phosphate. These are the pyrimidine class of compounds which are synthesized, and this uridine triphosphate happens in case of the RNA molecule. The pyrimidine synthesis happens in case of the RNA formation, but in case of the DNA formation, this orotidine monophosphate gets converted to thymidine monophosphate, TMP. Now the thymidine monophosphate again gets converted to thymidine triphosphate, TTP. So this is in case of DNA and this is in case of RNA. So this is what the de novo pathway of pyrimidine metabolism is, and in the case of the previous video I have shown that the purine metabolism is there in the de novo pathway only. All of those conversions, those huge conversions taking place by lowering the activation energy along with the energy taken or the energy that is granted by the conversion of ATP to ADP plus PI. Even in this case also, ATP to conversion of ATP plus PI, it's happening because it's very evident for the formation of stable nucleotides or mature nucleotides in the purine metabolism de novo as well as in the pyrimidine metabolism too. So this is how entire de novo pathway of pyrimidine metabolism happens, that is the synthesis of the pyrimidine class of nucleotides.